Good morning, students. Today we will be discussing about disease, which is basically associated with direct contact So, the direct contact disease, the first disease which we will be discussing, that is your Staphylococcus. Staphylococcus. Now, you know that the genus Staphylococcus contains pathogens of humans and animals. And staphylococci commonly infect skins and wound, may also cause pneumonia. Now, students, when I'm talking about the staphylococcus, you may be thinking that we are handling it in the lab. So, when we are handling it in the lab, so what we can do is that we can understand the importance of this particular organism. We can understand that what is this particular importance, why it is, whether it is dangerous, whether it is not, it could be easily handleable. So we need to understand, though we know that Staphylococcus falls under risk group 2, but still we need to understand the disease caused by them. So basically, they infect through skin. Through skin. They infect through skin, they infect through wounds. So they infect through skin, they infect through wounds, and they also cause pneumonia. Okay, so direct contact is from Staphylococcus aureus, and moreover direct contact disease, and it is basically a pathogen which basically infects skin, basically infect the wound, and also cause pneumonia. Okay, next we will move on. To the morphology of your Staphylococcus. So we will move on to the morphology of Staphylococcus species. So morphology of Staphylococcus is that it is non-sporulating. It doesn't produce spore. Non-sporulating. It is non-sporulating. It is gram positive. It is facultatively aerobic cocci. Coccus, the default shape of your bacteria, and it is 0.5 to 1.5. 0.5 to 1.5 micrometer in diameter. So these are the basic structure, the morphology of your Staphylococcus. As you know that you will see a bunch of grapes, right? Because this particular coccus grows in all the different planes, and so you get a bunch of grapes. It is gram-positive in nature. It is non-sporulating. It is facultatively aerobic, and the dimension is 0.5 to 1.5 micrometer. Irregular clump of cells. Next, we will move on to the various strains of Staphylococcus which is dispersed into the environment. So, first we will move on to Staphylococcus epidermidis. Staphylococcus epidermidis, a non-pigmented species usually found on the skin and mucous membrane. So it's a non-pigmented species. You will not get to see that it is producing any pigment, right? And you will find it diversely in your skin. Whereas Staphylococcus aureus Staphylococcus aureus is basically yellow pigmented species. So it is yellow pigmented species. It is yellow pigmented species, so it will give you an overall yellow pigment, right? And it is associated with the various human diseases. Epidemiology. Epidemiology and pathogenesis. Staphylococcus cause disease including the various types of disease like acne. It causes acne. It causes boils. It causes pimples. It causes pimples. It causes impetigo, pneumonia. So it's a multiple different type of disease which is basically being produced by Staphylococcus. Then your osteomyelitis, osteomyelitis, carditis, meningitis, arthritis. So many different types of disease is caused by Staphylococcus, and many of these disease are basically pyogenic in nature. Pyogenic in nature. Many of the disease is pyogenic in nature. That is, it is pus forming in nature. Okay. So healthy individuals are often carriers and resident of Staphylococci in the upper respiratory tract or skin, seldom cause disease. But if you understand that if there is a crack in the skin, then through that it can enter into the body and can cause many different types of disease. And it is also very important that the host is not immunocompromised. That is very important. Now, let's move on to the other virulence factors, which are basically produced by the Staphylococcus. So, you know that all Staphylococcus produce first important thing, that is your catalase. Catalase, that is an enzyme that converts hydrogen peroxide to water and oxygen. So catalase is not considered as a virulence factor, but catalase test distinguish Staphylococci from Streptococci, which do not produce catalase. So Staphylococcus produce catalase, but Streptococci do not produce catalase. Then we come to the virulence virulence factors. So in virulence factors, the first important virulence factor is the cytolytic toxin, which is basically produced by Staphylococcus, and this toxin is called as hemolysin. This toxin is called as hemolysin. Hemolysin may cause cell lysis in vivo and cause red blood cell lysis. These organisms are fastidious group of organisms. So they depend on something they depend this on blood to survive. So this is your hemolysin, and cause lysis in vivo and cause red blood cell lysis. So you will get to see different kinds of lysis in your blood agar plate. Blood agar plates. So they can lyse the RBC. This is one of the very important virulence factors in the Staphylococcus aureus. Next is the production of coagulase. S. aureus produces Coagulase. Coagulase. So coagulase is an enzyme that converts fibrin to fibrinogen. So fibrin to fibrinogen forming a localized clot. So they produced a localized clot. Okay. So within that clot they may hide from the immune system to prevent them from being phagocytosed. Next is the production of leukocidin. So there is another production, that is production of leukocidin, and this leukocidin basically destroys leukocytes. So we will have, so these are the different virulence factors of S. aureus. That is, you have hemolysin, you have coagulase enzyme, you have leukocidin by which it can cause disease, and there are many other proteolytic enzymes like hyaluronidase, fibrinolysin, lipase, ribonuclease, deoxyribonuclease, which is present in them. So for today's class we have discussed the occurrence, the morphology, and the virulence factor of Staphylococcus aureus. Thank you.